Hello and welcome back to Chapter 7. In this section, in Part 2 here, we're going to talk about gene expression, so we will address transcription and translation. So in gene expression, this is where the genetic material of the cell is going to be copied from DNA into messenger RNA, and so that's transcription. And then when messenger RNA meets up with a ribosome for protein synthesis, that will be translation. And transcription is really a very simple process compared to DNA replication or even compared to translation. In transcription, RNA polymerase is the enzyme that will initiate. So transcription, just like replication, will go through initiation, elongation, and termination. And RNA polymerase initiates transcription at a sequence in front of, just upstream of a gene, called a promoter. And most genes will have kind of two sections. They have this regulatory section that occurs just in front of the gene, and that regulatory region, you might say, is usually composed of a promoter and an operator. Now, the promoter is a sequence that is used for recognition. The promoter, just like a concert promoter or an event promoter would say, hey, this is where the event is, this is what time it's at, gives you all the information to be able to attend that event. Well, a promoter sequence in a gene, in gene expression, is the same thing. It's a promoter sequence saying, hey, over here, this is the gene for such and such. If you're such and such a transcription factor, you need to come over here. This is where you can get things started. And it's at that promoter sequence that RNA polymerase is going to recognize not only transcription factors unique to that particular gene for expression, but will also be activated by another protein called a sigma factor. And that sigma factor is required in order for expression to occur, in order for RNA polymerase to be activated and move forward towards transcription. Now, once it passes the promoter, it enters into another part of that regulatory region called the operator. And you can kind of think of the operator as the bouncer outside of a club. And the operator is a region that if a gene is going to be turned off, then a repressor might sit there, or a promoter, enhancer might come in. So the operator sequence is really kind of the gatekeeper of whether this moves forward or not, and is oftentimes where regulation, as we think of it in terms of repressing or enhancing, might actually occur. So once RNA polymerase gets activated by the sigma factor, it's pretty much given permission at the operator. It moves forward and it begins the transcription process. Now, in the transcription process, the RNA polymerase is going to complementary base pair RNA nucleotides to one of the DNA strands. Now, let's take a look at this diagram here. If the gene is here on this positive strand, the positive strand is the one that contains the gene, then RNA polymerase is going to complementary base pair to the negative strand. And this is because we want an actual copy of the gene. We want our RNA sequence to reflect this DNA sequence. And because RNA polymerase uses base pairing, if the positive strand is the gene, it's going to have to complementary base pair to the complement of the gene. Think mirror image of a mirror image. And this will then provide us with the correct sequence up here. So you see, this is the complement to the negative strand. If I just went directly off the gene and complementary base paired off the gene, I would get the wrong sequence. I would end up with the negative strand sequence in RNA language. So I'm trying to transcribe that gene. I need to make sure that I'm transcribing the correct strand. When RNA polymerase moves across and begins reading this gene, this DNA strand, it's only going to be able to add to the 3' end. Right, to the 3' end. So when it moves across for this gene, it's going to be moving in this direction, because every time it adds a nucleotide, the 3' end is going to be here. So it has to have an existing 3' for the incoming 5' carbon. Remember that sugar phosphate backbone. The RNA transcript is complementary to DNA, so it's that complementary base pairing like we saw in DNA replication. The same thing is occurring in transcription. And once the mRNA transcript is being made here in prokaryotes, when an RNA transcript is being made, ribosomes are going to attach and begin the translation process even before an mRNA transcript is completed. So it's polyribosomal. We have lots of ribosomes are going to be sitting on that mRNA transcript while transcription is still occurring. In eukaryotic cells, this process is separated spatially. So in eukaryotic cells, mRNA has to be processed. There's a lot of things that have to happen to it before it can enter into the cytoplasm and meet with the ribosome and begin the translation process. So for prokaryotes, transcription and translation are simultaneous. In eukaryotes, they are two very separate processes. So here's transcription. Here is our RNA polymerase. Here is our sigma factor, and they're showing you here, this is the promoter sequence in front of the gene. And here RNA polymerase is going to move to the right. It is going to be complementary base pairing off of this top strand. Right? So it's moving from 5 to 3, building 3 to 5. And you can see here it's complementary base pairing on that top strand there. Now notice, unlike in DNA replication, in RNA polymerase, the RNA transcript is coming off of RNA polymerase, and the DNA is, sorry about that, the DNA is zipping right up behind it. So the DNA just kind of opens just enough for that RNA polymerase to move through. And eventually, this is the elongation process. So here we have initiation, right? Oh, they have it labeled there for you, so we'll just do it again. So here's initiation, RNA polymerase being activated by sigma factor at the promoter sequence. Once RNA polymerase is activated and begins creating that mRNA transcript, we're now in the elongation stage. And this is just straight up complementary base pairing. Eventually, it's going to reach a sequence down here called a termination sequence. And that's referred to as a terminator. And at this point, the RNA polymerase just kind of dissociates or falls off, leaves the DNA transcript and releases the mRNA transcript. This would be the 3' end. Releases this mRNA transcript for ribosomes to complete the transcription process. So that would be termination. Now, in initiation, just to kind of review real quick, remember we've got RNA polymerase coming in here, and it's going to bind at this promoter sequence with its sigma factor. Now, in this case, if my gene is on the top strand, then this one is going to move across this way. And here, if my gene is on the top strand, it's going to have to move, or it's going to move in this direction. So depending on the orientation of the promoter, so which way is the promoter facing, that's going to determine the directionality of RNA polymerase. And so that is a specific sequence, and sigma factors and other transcription factors are going to recognize not only the sequence, but the direction of that sequence. So again, initiation is sigma factor and RNA polymerase recognizing the promoter. Elongation is the actual synthesis of the messenger RNA transcript, and termination results in a hairpin loop at the end. Hairpin loop is when the RNA transcript tends to self-bind to itself like that, and it creates this kind of loop. And when it creates this loop, it disrupts the binding process inside of RNA polymerase, and RNA polymerase is unable to continue. So that's why it dissociates. So let's talk real quick about translation. Now, in translation, we have to decode the mRNA message that's coming from DNA, and ribosomes are responsible for that. But in conjunction with ribosomes, we're also going to have tRNA, transfer RNA. And transfer RNA is a very important player because transfer RNA is who brings in the actual amino acids. So that step between nucleotides and protein occurs within the ribosome. tRNAs are that bridge that occur between them. Translation requires ribosome large and small subunits containing ribosomal RNA. It also requires what we call charged tRNA. This would be a transfer RNA with an amino acid attached to it. And then, of course, we need messenger RNA. We need the actual message to be translated. Again, translation occurs simultaneously, synchronously with transcription in prokaryotes, but not in eukaryotes. Just want to show you here real quick the structure of transfer RNA since it's such a key player. It may not make a lot of sense to you right at this moment, but as we move through translation, this structure will start to make some more sense. Right here, these little loops are what I was talking about when I was talking about a hairpin loop, where this is actually a single-stranded RNA molecule that has complementary self-base paired. It has base paired to its own strand, causing this bending or shape. And when this occurs, it can only go so far. And when it gets to towards the end of where a bend is occurring, these bases here can't really reach each other. So they're kind of left in this hairpin loop-like structure. But they're attracted to each other, and so they're facing inwards in this loop so that there's no random binding. Same thing is happening in this loop over here as well. So those are bases that are facing inwards. But in our tRNA, at the opposite end of the amino acid site, at the very opposite end, in this turn here, when this loop occurs, the RNA gets twisted so that 3 of the bases are sticking outwards instead of inwards. So these guys might be inwards, but there's a twist that occurs somewhere in here that causes 3 of these to kind of flip out and be facing out. This is referred to as an anticodon. And the anticodon is going to complementary base pair, right? It's 3 bases. It's going to complementary base pair to 3 bases on the messenger RNA. And on the messenger RNA, those 3 bases are called a codon. So the anticodon of tRNA is going to be complementary to a codon on the messenger RNA. And on this messenger RNA, the next 3 bases are the next codon. These 3 bases are a codon. So a codon is a triplet of bases that will be complementary base paired, self-base paired to, I'm sorry, not self-base paired, but complementary base paired to the messenger RNA transcript during the process of translation. And every time one of these tRNAs, the anticodon, complementary base pairs all 3 of its bases to the codon being presented, the amino acid of which it is carrying is going to be incorporated into the primary sequence of the protein. So let's take a look at our genetic code. So remember, we said that our tRNA had an anticodon, and the anticodon is going to match 3 bases in the mRNA transcript that are called codons. And the codons, or the genetic code, has been decoded. And we know what each of these codons represent. Now, there are 20 essential amino acids required in most organisms. And it's usually pretty much the same 20 amino acids. We're not going to go into any major differences. But for the most part, we have 20 essential amino acids. And these are all of the amino acids required to make up proteins for an organism cell for a cell to function. Now, when we look at these codons, we have a first position, a second position, and a third position. Now, one thing I want you to notice is this third position is sometimes called a wobble, or a wobble position. Wobble base, it's called a few different things. But it's called a wobble because hydrogen bonding occurs pretty strongly between the first and second bases, right? So we have good hydrogen bonding here. We're talking anticodon to codon. But it's happening so quickly that this third base, it'll bind, but it's so fast that if it doesn't completely bind to that third base, the reaction will still move forward. So what we see here is we see our first position. When you read a table here, here's our first position, right? A U, a C, an A, and a G. And this is on the mRNA transcript, okay? We're reading codons. And then we have the second position, a U, a C, an A, and a G. And then each individual row is the third position, or wobble position. And in this third position, you'll notice that most of our amino acids, let's see, leucine, serine, proline, threonine, alanine, valine, most of these, right, arginine, glycine, most of these have 4 different codons, right? So this one has 2, excuse me. So here I have 4 different codons. All 4 of these codons have the same first position, they're all C's, the same second position, all U's, but then any of the bases can be in the third position. And this allows flexibility in the genetic code because of how that base pairing occurs between the anticodon and these codons that are presented here. We're going to see that there needs to be some flexibility. And since there's only 20 amino acids and we have 64 codons, we can afford some flexibility in some of these that that third base could be any of the 4 RNA bases and it would still be the correct protein. So this helps to prevent mutations from being too prevalent. Right? We might have what we might call a silent mutation where the wild type gene is a U and the mutation is a C in the third position, but it's a silent mutation because it doesn't matter what base is in that third position. We're still encoding for the same amino acid. So it allows this dynamic type of flexibility in the genetic code. So reading a genetic table is going to be important. You're going to need to be able to read one of these in your exam. So make sure that you are familiar with that, with how to read one of these so that you can translate an mRNA transcript. So looking at this genetic table here, our genetic code, here you can see we have our tRNAs coming in complementary base pairing to that mRNA sequence, and as it does it, these amino acids are going to get added in a very specific order, and that's the primary sequence of a protein. When you learned about protein folding in your anatomy classes, we had primary, secondary, tertiary, and quaternary. And in primary sequence, that's the sequence of amino acids. And here is how that sequence of amino acids is determined. The ribosome will read the mRNA transcript. It starts at the 5' end, and it begins reading that mRNA transcript and starts bringing in anticodons to try and complementary base pair those anticodons to the codons being presented in the mRNA. As it does that, if it matches, the tRNA will then begin stacking these amino acids to create the polypeptide, and we'll look at that in a second. So here we have our codons. Our codons are being presented, and they're being presented 3 at a time. And so these amino acids are going to be put together in this specific order. So this is our primary sequence of a protein, right? Primary structure. And in learning about protein folding, we know that DNA determines the primary sequence of a protein, which is the sequence of amino acids. Okay? So during translation, ribosomes are responsible for initiating this translation, and translation, just like replication and transcription, has initiation, elongation, and termination. So these 3 steps are important in replication, transcription, and translation. And in initiation, we start with the ribosome binding to the mRNA transcript. It does this at the 5' end, and it does it at a site called the ribosome binding site. And as the ribosome begins reading, remember, ribosomes are proteins with mRNA, ribosomes are proteins with ribosomal RNA kind of wound throughout the protein, right? Intertwined with that protein. And they are part of the ribosome itself. Ribosomal RNA is necessary for a ribosome to function. And one of the primary functions of that ribosomal RNA is to recognize the 5' end of an mRNA transcript and to recognize the ribosomal binding site. And the ribosomal binding site is known as the Shine-Dalgarno site. And the Shine-Dalgarno site is just a short little nucleotide sequence. It's very conserved from one species of bacteria to another. Remember, we're talking about bacteria here. And that sequence is common from one organism to the next. And in that sequence is kind of like raises the flag and says, okay, I know you entered here at your 5' end, but until I reach that Shine-Dalgarno sequence, I'm not going to begin translation. But once the Shine-Dalgarno sequence is reached, the very first start codon after the Shine-Dalgarno sequence is where translation, actually bringing in tRNAs, is going to begin. And this is important for reading, for setting the reading frame. If the ribosome does not set the proper reading frame, it's going to put the wrong amino acids in the wrong sequence, and the protein is not going to be functional. So that Shine-Dalgarno sequence is necessary to say, hey, get ready, because the next AUG you come across is where you need to start translating. And that initiates processes in the ribosome to ensure that it is ready to begin that base pairing at the next AUG or start codon. After that initial AUG tRNA complement binds, we're now moving into the elongation stage. And so that first AUG always encodes for formylmethionine. It's called FMET in bacteria. In eukaryotic cells, it's just methionine. So it'll start with that formylmethionine amino acid. Once it enters into the elongation stage, the transfer RNA with its complement anticodon to the mRNA is presented, and it binds in a site of the ribosome called the A site. The ribosome has 3 binding sites. I'll just draw them for you here. We have A, the A site, the acetyl site. We have P, which is the peptidyl site, and E is for exit or ejection. And that tRNA is going to get presented in the A site as our mRNA moves through and has our 3 bases here. And if the tRNA, right, he has his amino acid here, if the tRNA matches, then everything shifts over 3 bases. So now I have these 3 bases here, and I got this guy with his amino acid on this codon that he base paired to, and the next codon gets presented in the A site. The previous tRNA is in the P site, and once the tRNA in the A site complementary binds to the mRNA that's being presented, to the codon being presented, this causes a conformational change in the ribosome. And using some GTP, this guy here in the P site is going to hand off his polypeptide here. The peptide bond occurs in between. Everybody shifts over another 3 bases. This tRNA will now get ejected because it's no longer charged. It has to go out into the cytoplasm to go get charged. This guy here is going to move over, and now he's holding on to 2. The next one gets presented, it binds, and then the process repeats again. So that in this P site, we're going to see this constant building or holding on to the polypeptide. So the polypeptide will grow and be held onto in the P site. And that P site is a peptidyl site for creating a peptide bond between the existing polypeptide chain and the incoming next amino acid. And all of those amino acids are being presented by the tRNAs, and the tRNAs ensure that it's the correct amino acid by complementary base pairing to the codon being presented by the messenger RNA. All right, my video is not working for some reason. Let me try changing my pointer here. It might be because I'm on a pen. Nope. Well, we'll just draw on it then. Fix our pointer here. There we go. Sorry about that. So again, I just kind of want to show you here how this is moving across. We've got our growing polypeptide. Okay, here's our growing polypeptide. Here's our incoming tRNA. This is the A site, this is the P site, and of course this is the E site. And what's going to happen is that this tRNA is going to kind of come over here. This guy's going to hand off that blue amino acid and create a peptide bond between the blue amino acid and the pink one. Everybody will shift over 3 bases. This codon will be presented next. This one moves into here. This guy will move into the E site and be uncharged, be ejected out of the ribosome to go pick up another amino acid. And then this pink one will be holding on to the entire polypeptide chain. And again, that process will continue to repeat itself until the ribosome reaches the termination sequence in the mRNA molecule. So this is just kind of showing you how RNA polymerase and ribosomes are working together for transcription and translation to occur at the same time. So it's called a polyribosome because there are multiple ribosomes. Of course, this guy attached first, so he's got the longest polypeptide. termination will occur at the stop codon. And when the stop codon is reached, the tRNAs don't carry in, there's not really a tRNA for a stop codon. So there's no anticodon to match the stop codon. It's actually a termination, it's called a termination factor. It's a protein that will match and say, nope, we're done here. And then that allows the polypeptide to be released. The ribosome will fall apart, the polypeptide will be released, and it'll go into post-translational modification. Now, post-translational modification includes assistance in folding, because now we've created the primary sequence of the protein, but we need to ensure that alpha helices, beta sheets, and then, of course, tertiary structure is correct. So there are lots of proteins called chaperones, and these chaperone proteins will basically encase that primary sequence, that polypeptide sequence, and ensure that those bases, that those functional groups, those R groups on the amino acids interact correctly so that the protein folds into the proper three-dimensional sequence. There's also a, for proteins that are going to be secreted or exit out of the cell itself, they oftentimes require tagging. They have to be tagged. And so this is known as signal sequence recognition. And in this process, proteins, including chaperones, will add molecular tags, whatever those tags may be. They'll add molecular tags so that the plasma membrane or cell membrane recognizes these proteins as being excreted or released. Usually it includes some type of hydrophobic molecule. All right, so my videos are not working here. I will upload these videos. We watch them in class, and I'll upload links to these videos for you in D2L. So there are some major differences between prokaryotic and eukaryotic transcription and translation. In prokaryotes, the messenger RNA does not get processed following transcription, while in eukaryotes, there's a lot of processing that occurs. In that processing, eukaryotic mRNA will receive a 5' cap, a 3' poly A tail. Also, eukaryotic mRNA contains what are called introns and exons. These are regions of a transcript. Some introns will not be expressed. They're going to be spliced out, and then exons are going to be brought together. Prokaryotic cells don't have any of that. Because that transcription and translation is simultaneous, the mRNA transcript is going to just strictly be all of the codons in the correct order. There'll be no 5' cap, no poly A tail, and there's certainly not any introns or anything in the prokaryotic. Translation and transcription occur simultaneously, where in eukaryotes it is spatially separated. mRNA in prokaryotes is polycistronic, where in mRNA it is monocistronic. So polycistronic, just to remind you, means that more than one gene can be in that mRNA sequence. Because bacteria have operons. Oftentimes encode for 2 or more genes with one regulatory region. So all 2 or 3 or 4 of those genes are all going to be transcribed and translated together to create one really large mRNA molecule, and the ribosome is going to encode all of the proteins in that mRNA in one shot. Whereas with eukaryotes, once our mRNA transcript is made, it's only going to make one protein. We're not going to have multiple proteins on the same mRNA transcript. So polycistronic is multiple transcription genes or multiple proteins being made from a single mRNA transcript, and monocistronic is one protein being made from a mRNA transcript. And finally, in prokaryotes, translation begins after that Shine-Dalgarno, that first AUG, whereas in eukaryotes, it's actually the first AUG. There is something similar to a Shine-Dalgarno that is seen in eukaryotic DNA, and sometimes that might be organismal specific. So that completes our lecture on transcription and translation. We covered a lot of this in class, and this is just kind of a recap of what we covered in class. Again, I will provide links to those videos for you in D2L, since they weren't working in the PowerPoint today. So I will see you guys in class. Have a good day.